I'd like to thank everybody first for coming to the mini symposium. So the fact that we have such a great turnout, I think this is thanks to many, many people as well as PIs who are willing to let the precious students to come to our symposium or mini symposium. So my name is Charles Lai, and as you can see right here, that's my name, or my Chinese name would be Lai Pingguang. But given that rumor has it that I speak Mandarin really poorly, so I'll use English instead. So I'm currently Assistant Research Fellow at Institute of Atomic and Molecular Sciences, as you heard from my director, Director Chen earlier. I previously served at NTHU, so I also had the privilege to work as separate colleagues. So without further ado, I was given the honorable task of doing the test, the past, the present, and the future of EV research and application, and it turned out that I'm the most junior guy here. So that kind of doesn't make sense, right? That should be a really junior guy's job. But so if I say anything wrong, please forgive me. Now, I've been working on vesicle for the past 7 to 8 years, and I think I'll start with an analogy. If you're new to this field, what are vesicles? Now think about network in the real life that we live in today's world, that we're surrounded by a tons of network in the world that we live in. So we do have, for example, the direct communication that's formed by cables, by wires. Do you know that your internet is actually connected between continents by sea cables? Did you know that? It's not through satellite. That's actually the most stable connection. How about wireless? Have you lived through the age without Wi-Fi? I had. It sucked. Okay, seriously. Back in the days when I was a kid, when I want to use internet, I had to use a telephone, I had to use a modem, and I had to go dee, ta, 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 and finally connect. And the speed I got the fastest was 56K. Okay, that's really, really slow. Nowadays it's different. You have like 3G, 4G, LTE, and now we have 5G coming up hopefully in the next couple of years. At least I know the States is starting that next year. So with that being said, what about the networks in our body? Do we know how cells connect to each other directly? Now, coincidentally enough, I used to work on something called gap junctions. So cells actually touch one another to form physical conduit, okay, and they can actually exchange information. And this was actually my former PI's lab, Dr. Christian Naus. We actually see the exchange of material between cells. That includes nucleic acid, calcium, and etc. And it was actually very immediate, sort of like the internet cable that we have. How about indirect? How do cells actually connect or talk to one another? Now, I believe that most of us are really, really familiar with protein receptor. Those are the most common form of indirect communication to our knowledge. But it was until the advent of vesicles that came out that completely changed the horizon of our understanding on cell-to-cell communication. Coming back to the analogy, this is what we knew in our world. Going a little bit back in history, around the 1500s when Leonardo da Vinci was still alive, this is what he drew about our human body. To his knowledge, this is how we are connected. We have blood vessels, these are our organs. Well, he didn't go into the brain, I guess. But this is what we knew at that time, that cells need to contact one another, or at least through the different vessels, be it lymphatic or blood, that this is how we actually communicate. And now we are in a new era, sort of like the Columbus era, that he's trying to explore the new continents that's still unknown to us. So we're really in a very, very interesting age where we're trying to explore something that was completely unknown to us. Therefore, there's a lot of things and opportunities that people can jump onto. Now, a little bit of history. Now, this is really brief history, so forgive me if I really oversimplify it, but it was in 1983 when vesicle was first detected from reticulocytes, basically immature blood cells. And then what they found was that there are sizes around 50 nanometer that's being released by these reticulocytes, and then they actually carry transferrin receptors on them. And then people started to use the term exosomes around 1981, even before this paper was published, to coin vesicles that are released by the cells. Then there was a huge, long time of void where nothing was published. Okay, I don't know what people were doing at that time. Okay, maybe they had a really fun pot or whatever that makes them happy. It was only until around 1996 and 1998 that Raposo found that B lymphocytes releases vesicles. She only used vesicles as a term on her paper at that time. And then Zitvogel's group actually used the term exosomes for vesicles released by dendritic cells. Then coming to the past and the present connection, people started calling various type of vesicles, such as Dr. Guo mentioned, there are many, many names. So between the age of 2000 and 2018, various types of vesicles have been found in humans between normal and disease cells, so cancer, Alzheimer's disease, Parkinson's disease, you name it. Any type of disease cells and normal cells have been compared on their vesicle phenotypes and status. They have found it in semen, blood, breast milk, amniotic ascites, cerebrospinal fluids. Okay, oh, I had a typo here. And then further to that, people have been finding vesicles from plants, as Dr. Guo briefly mentioned and discussed, and more importantly, bacteria. Now, the possibility is literally endless. Like yeast, they also release vesicle. Anything that you kind of think about have vesicles being released. Now, vesicle is only the carrier, right? But think about the amount of cargo that they package into. And if you relate that to the world that we live in right now, it's actually very similar. That we're really entwined by all these vesicles, and the information that's being exchanged is extremely dynamic. To decipher those is your job, and as well as my job, because that's how I keep believing. But you know, this is actually very exciting. How do you actually decipher such a complicated network? And in fact, people have found that in the sea, that there's a huge biomass consisted of vesicle. This has been published a few years ago. So the sheer amount of information that's being exchanged in the world that we live in by vesicle is enormous. Now, coming to the present, we now call them extracellular vesicle because there were so many names. Because, for example, people coin the term exosome nowadays just by their size, ranging from around 40 to 100 nanometer. Shed microvesicles is probably another one that you hear a little bit more. They can be up to a micron. Apoptotic bodies or blebs, they can be up to 4,000 nanometer. And some people believe is the Visio, they actually suggest cancers can release vesicles up to 10 micron. So that's really, really big. So to my understanding, without offending any scholars, people started to use the term extracellular vesicle to collectively term this vesicle so that we don't argue against, you know, ectosome, exosome, blah, blah, blah. It's a little bit too complicated. And another point to that is that we, at least to my knowledge, we don't have a technology to specifically isolate a single subtype of vesicle. And in fact, if you really want to use the term exosome, according to Dr. Terry Clotil's recent study, you need to have 3 markers to actually call it exosome. So the criteria is actually quite stringent. Now, I know that we have many, many great talks today, but as molecular imaging researcher, seeing is actually believing. Now, I just really want to show you this to kind of have appreciation of what vesicles are. This is a 2-photon imaging setup that we did back in when I was younger in Boston with a collaboration with Dr. Torsten Mempo as well as my mentor, Dr. Sandra Brakefield. What we did is that we labeled all the cells. So in this case, this is thymoma cell. You see a lot of big green cells. And then we labeled them with a reporter. We developed reporters to label all the membranes on the cells and later vesicles. And then we implanted into the dorsal skin fold chamber right here. And then we allow these tumor to grow for about a week. Then we did multiphoton microscopy. And on the left right here is actually what we could image. So you see a lot of cells, but you also see many, many tiny little dots floating between the intercellular space. And then if we actually enlarge it to the top right here, we can actually see that the vesicle seems to be released. Be it shedding or be it, you know, exosomes, they are being released. And further, if we look at the intercellular space, so this void right here, vesicles are dynamically floating around, but some of them seems to be traveling in groups. Now, this may not surprise you much, but think about this. This is only over a span of 30 minutes of imaging. And these are from cancer cells, which we know that they actually release more than normal cells. But to our knowledge today, all types of cells release vesicle. All normal cells release vesicle. Hopefully you don't have cancer. But this is happening 24/7, 365 days a year. Imagine the amount of vesicle that's been released by both normal and disease cell. Imagine the amount of cargo that's being trafficked, the amount of message that's being exchanged. That's really exciting. And not only that, we haven't even talked about the bacteria, the human interaction, and also what you eat, right? So I think this is very exciting. So if we look into this, how about the present to the future on EV research? So I really simplify this down that essentially you have the EVs. This is EM to show that the very canonical lipid bilayer structure. Basically, this is what makes vesicle exciting, is that they are a very efficient carrier that's more endogenous, that can have both protein, RNA, as well as DNA. And I think right now a lot of people are trying to study EV biology in the sense that how are vesicles released? How are they regulated? Can we actually reverse engineer that? As well as how are they involved in development? That have been found because vesicles are involved in Wnt signaling and other stuff in terms of their posterior anterior positioning. Normal and diseases. This is probably the most exciting part that people are looking at. Vesicle has been involved in any disease that you can think about, ranging from infection to, I don't know, stroke, and then like anything that we can think about, vesicles almost always related. And I think another really exciting field is, since they can actually carry all these cargoes, they are actually a very good vehicle to preserve all these cargo from readily degraded in the blood or the fluid. And then based on that, in fact, for example, Dr. Takahiro Ochiya just mentioned, they could actually diagnose cancer. And then, as you know, that non-invasive diagnosis is really hot right now. And vesicle is actually one of the targets that people are trying to isolate so that they can extrapolate the microRNA to find the signature and then correlate that to the disease status. Now, therapy is another field that people are trying to go into, is that if they can use this as a gene therapy vehicle or drug delivery, as Dr. Go had published, this would be very advantageous than the more quote-unquote artificial synthesized vehicles such as liposome that people used to work on. And then, not only that, can you potentially load your cargo of preference, you could potentially target them because, for example, Dr. Shen Tongno's lab, and then in collaboration with Dr. David Leiden, they have found that specific integrin can actually target vesicle to specific organs. So that's really exciting if we know how to reverse engineer the vesicle properties. And how about regenerative medicine? Everybody's getting old, including myself. How do you actually prevent aging? How do you actually regenerate your tissues? Can you actually potentially use vesicle to help with that? And in fact, I know Dr. Sai-Kiang Lim from Singapore, A-Star, they're also working massively on producing mesenchymal stem cell derived vesicle for therapy or regenerative medicine. And of course, I know that many other people are working on that. So it's a very exciting field, given especially for Asia or South Asian Pacific countries, that we are going into an aging population, especially Taiwan, and I know that Japan has already hit into an Asian population. So not only that, I think this is a really exciting part, consumer products. Can you actually make vesicle for consumer product? And you may be like, meh, no, this is a huge market. For example, cosmetics. For example, probiotics. Those kind of things. That's what a lot of people are really looking into, because vesicle may be a really endogenous and potentially beneficial vehicle to be more, supplementary to our human diets. Now, to sum it up a little bit, there are many societies for vesicle in the world that we live in today. One thing that I think that stood out the most that you hear again and again is the International Society for or of Extracellular Vesicles. We call them ICEV. To my knowledge, I think the initial pre-meeting was in Paris. Then this was followed by the one in Gothenburg, Sweden in 2012. And then they have many meetings later on, but they usually hold their meeting, like consecutively between North America as well as Europe. But I think they had it 2 times in Rotterdam or something like that. And then they also had it in Boston. And then they kind of just exchanged between them year after year. Now, there are many, many other ones. For example, I don't know how to pronounce it, Australasia, EV. There's also the American Society for Extracellular Vesicles, Korean Society for Extracellular Vesicles. This is by alphabetical order. So, except the first one, because this was one of the earlier ones. And then Japanese Society for Extracellular Vesicles, and also the one in Singapore. So there are many, and I was just checking this morning. There's also one in German. There's another one in Netherlands. So every country is almost hosting one vesicle society for themselves because everybody is really interested in that. Now, how about Taiwan? So I think we're really glad to finally have our society formed today, if you're here a little bit earlier enough. And since I grew up in Canada when I was a kid, though I'm MIT, made in Taiwan, that I really like this quote from a radio show from the Canadian Broadcasting Company. It says, "We may not be big, but we're small." And I thought that kind of fittingly described our society today, that we're kind of small, but I'm actually a little bit surprised. We have a really good turnout today. But we're hoping that from this place where we start, that with the Taiwanese Society for Extracellular Vesicles, that we have a couple aims, is that we can try to advocate and educate on vesicle research. And I think the term education is a little bit humbling. It's basically that we actually ran into more problems than you did before, so that we're hoping to share our experience with you so we don't make the same mistake. So I think that's something that we want to do. But on top of it, I think this is something already happening, that we're hoping to establish more and more international and local collaborations. And beyond that, we're hoping to foster the collaboration between academic, the medical field, and more importantly, also the industrial field. I think that's what could make the vesicle research even more dynamic and more applicable from our basic research perspective. How about upcoming events? I think that's something that Dr. Shintano mentioned, is that we do have an EV workshop at the 2018 Annual Biomedical Sciences Conference, or 生医年会. And then this is we really appreciate Dr. Wu Yingqin's support, as well as the 中华民国细胞及分子生物学学会. So it's really twisting the tongue, but the CSCMB. So this will happen in March the 25th. And then Dr. Go will be there as well. And then we're hoping to invite really a lot more great speakers to come. And then without further ado, I would really like to thank all the sponsors that make today's mini symposium possible. That includes the Taiwan Breast Cancer Foundation from Dr. Zhang Jingjie and Dr. Guo, the Industrial Technology Research Institute, the Bio Group, the Jing Online, which is our media sponsor that really put a lot of work to make. Actually, the fact that you know how to come here is probably through them. Yeah, because it's really important that we spread the news out. And last but not least, because I work here, I need to sort of praise myself that thanks to the support of Academia Sinica, as well as this Institute of Atomic and Molecular Sciences, to make all these possible. And then if you're trainees, I would highly encourage you to talk to us. I think many PIs here are really excited to work on this, but we lack the people. I'm not telling you I'm telling you the truth. This is not fake news. This is the actual news. We really need new blood. We really need your excitement. We're really excited about this. This is a booming field that we hope you can come and participate, to share experiences, and then to help you, prosper. This is essentially what Dr. Shen and I have in mind to establish this society. So thank you very much for coming again, and then see you soon.